Okay. When I'm going to do this.
Okay, let's go ahead and get started now. We are going to take a look at the ear and the eyeball in detail, and then the brain. So I'd like to go ahead and start. Anybody have any questions on anything regarding this? Okay. So let's go into, let me go bring it up. Okay, let me now bring this up so everybody can see it and figure out what we're doing. Share screen. There we go. So I know you wouldn't have told me for a half an hour that I hadn't shared the screen. Yeah. So, okay, how do our ears work? The ears are designed to collect pressure waves. Pressure waves moving through the air. That's what they do. The pressure waves are formed anytime a sound is made. It's vibrations in the air. The pinna, the outer part of the ear, is designed to collect those pressure waves. You know, it's the original hearing aid is the pinna, and the pressure waves are caught by the pinna and sent down the auditory canal right here. This is the pinna out here. And if we look at our why isn't it? There it is. Okay. There we go. We look at the pinna. This very large structure right here is the pinna. It's like a funnel, if you will, to collect sound waves, so we can then pass them down into the ear canal here. So it's the original, which you consider the original hearing aid. People that have difficulty hearing will often cup their hand around their ear, around the pinna, just to try and hear a little bit better by catching. Didn't say anything about talking better. Catching sound waves or vibrations through here. Now from the pinna, we go down the auditory canal and we reach the tympanic membrane, the eardrum. The pressure waves cause the tympanic membrane to vibrate, and those vibrations then are transferred through the tympanic membrane into the middle ear using a series of bones that we call the malleus, the incus, and the stapes, or we've learned it when we were kids, it's the hammer, the anvil, and the stirrup, because the stapes looks like a stirrup. And so that is the middle ear. Now all we've done is take the vibrations that are the pressure waves coming into the ear and move them down to cause the tympanic membrane to vibrate, which vibrates these bones of the middle ear. Now the stapes connects to the inner ear. There is a structure in the inner ear called the vestibule, and it's filled with fluid. And the vibrations that are delivered to the vestibule through a structure known as the oval window. The stapes butts up on the oval window and allows its vibrations to go into the vestibule. Now, the vestibule does double duty. It connects to the semicircular canals, which allow us to deal with balance and equilibrium, and it connects to what's known as the cochlea, which is a structure that looks a lot like a snail. And so if we take a look here at our model again, we can see here is the ear canal, or the auditory canal here. Out here is the pinna, and we get into here and we can see the location of the bones of the middle ear. You know, this is the outer ear out here, and these are the ossicles, the malleus, the incus, and then we end up with the stapes right here. The stapes is a stirrup-shaped bone, and it connects to the vestibule. This structure here is the vestibule, and there's a structure right here called the oval window where the stapes butts up to it. Now this structure here, the vestibule, leads to either the semicircular canals, which are these, and you can see they run in 3 different planes, 2 of which are vertical and 1 is horizontal. And then over on the other side, we have the cochlea, which looks a lot like a snail. And it's the cochlea where we actually detect, where we are going to generate action potentials. Because the vibrations that come in through the oval window are transferred over to the cochlea, where they stimulate hair cells. The hair cells vibrate. They are mechanoreceptors. If they can hit threshold, then they're going to generate an action potential, which will move down the auditory branch of this nerve. This nerve is both balance and sound. You know, part of it's feeding from the semicircular canals, and the other part is for sound. And if we get an action potential, it will go up to the brain. Now, the brain is going to tell us where and what we're hearing. The ear has no idea. The ear can't tell us anything. All the ear can do is collect sensations that we can convert to action potentials. And remember, every action potential looks just like any other action potential. They're all the same. It's the area of the brain that interprets them as to what they are. You know, that the sound waves are converted to an action potential and sent to the brain, to the portion of the brain that interprets that. It's not going to go anywhere else, even though they all look the same. You know, the action potential from the optic nerve looks just like one from the olfactory nerve or the auditory nerve, but it's where they get interpreted. And so they'll go on up into the brain through this very large nerve right here. Now let's get back to, okay, so inside the cochlea are very tiny hair cells suspended in fluid. The hair cells will vibrate. They will start moving when the vibrations are transferred to them from the stapes through the oval window into the vestibule, into the cochlea. When they vibrate, that's when they function as mechanoreceptors, and they will generate a graded potential that may hit threshold. And if they do, well, then great. We have an action potential that we may or may not be able to interpret as sound. So, and you know, the ability to hear things in all different ranges depends on whether or not we're getting graded potentials that hit threshold. You know, because we all have different ranges of hearing. As we age, we lose more of the high ends. We may lose the low ends. When we're younger, when we're children, we can hear everything. We can hear the highs and the lows. As we age, though, we start to lose different ends of the auditory spectrum. Work environments may change that. If you work in a very loud environment constantly, you may lose the low end sounds, or you may lose the high end sounds. It just depends. Or you may not lose anything. You may be just fine. So, okay, now let's move on here. And there we go. And now, so the parts of the ear, the external ear, I've showed you the pinna. The pinna is just a funnel designed to collect the pressure waves. Early hearing aids were built on that. They were a large horn. Sometimes they were just like an animal horn carved out and stuck in your ear, and you collect the sound waves. Sometimes they were very large horns, handmade out of wood or something. You carry it around and stick it in your ear so you could catch the sound waves. It worked to a point. It worked when you were dealing with what we call conduction deafness, because it amplified this. It didn't amplify the sound, but you caught more of the sound waves, so you'd have a better chance of hearing it. So the outer ear is everything is the pinna up to the tympanic membrane. There are wax glands there inside this canal called the ceruminous glands. The wax is designed to trap dust and dirt particles and things like that, and insects and other unpleasant things to try and keep the ear canal clean, to keep it unobstructed so nothing gets back towards the tympanic membrane. On the other side of the tympanic membrane, you have the middle ear. You have the malleus, the incus, and the stapes, and you have the eustachian tube, or the auditory tube. This is what we use to equalize pressure between the middle ear and the nasal cavity. That eustachian tube extends from the middle ear to the back of the nasal cavity. The back wall of the nasal cavity is where the opening to the eustachian tube is located. When your ears pop, we are equalizing the outside air pressure with the pressure of the air in the middle ear. And we've all experienced that at some point. And then within the inner ear, we have the cochlea and the auditory nerve. We have the semicircular canals that deal with balance and equilibrium. And why did it do that? Okay. Now, so there's our ear in all its glory. You can see the pinna, sometimes called the auricle. Pressure waves come in here, vibrate the tympanic membrane. The vibrations go down the malleus, the incus, and the stapes. The oval window is right there where that butts up against the vestibule. This round structure here is the vestibule. Over here is the cochlea. We have the auditory nerve is actually part vestibular nerve and part cochlear nerve. So we call it the vestibulocochlear nerve. It's one of the cranial nerves. It's number 8 on the cranial nerves. And each branch sends signals, from their own, graded action potential. So semicircular canals tell us about balance and equilibrium, and the cochlea sends sounds. So, and here is the eustachian tube that goes from here to the nasopharynx, which is essentially in the back wall of the nasal cavity. In fact, I think, yes, you can see it right here. This structure there's our nasal cavity right here, and this structure located all the way in the back wall of the nasal cavity is the opening to the eustachian tube that leads into the ear. And so that's what we use to equalize our air pressure. Okay, and so, that's the tubes that we always hear about, having tubes replaced, tubes inserted in children. It is a perfect passageway for bacterial infection to come from the nasal cavity right into the inner ear for inner ear infections. So now, there are the ossicles, the bones of the middle ear, the malleus, the incus, and the stapes. And of course, the action is delivered then at the stapes through the oval window, which again, they're not showing here, but it's on the other side of that structure. Note that the bone here is spongy bone. All that bone around that is spongy bone, just like on our model here, it's spongy bone. And this is ideal for sound transmission. sound will pass through that spongy bone to get to the inner ear. So that's an alternative for getting sound to the inner ear. When we have a patient with a cochlear implant that's usually embedded somewhere in the mastoid process region and amplifies sound that pass into the inner ear. Very, very effective way of sound transmission. Not as effective as getting straight in through the auditory canal, but it still works. Now, it only helps if we're dealing with conduction issues, not nerve damage. Any kind of hearing aid, any kind of cochlear implant, all of these only work very well if we're dealing with a conduction issue. So, okay, so here is our ear model. It's not this one, this is a smaller model. It's only about 1 foot long instead of 3 feet long or whatever. So, excuse me, the pinna, the auditory canal, tympanic membrane, ossicles, eustachian tube or auditory tube, or whatever this word is, pharyngotympanic tube, cochlea, shaped like a snail. Now these are the semicircular canals. They are also filled with fluid and filled with hair cells. Movement of our head causes the fluid inside the canals to slosh around. So whichever way we turn our head, excuse me, whichever way we turn our head, the fluid inside these canals is going to slosh around. The hairs are going to move, and they're going to send signals to the brain, to the area dealing with equilibrium and balance, and tell us that our head's moving or that our body is turning. You can be in a car with your eyes shut, hopefully not driving, and when the car accelerates, when the car slows down, when we go around a curve, because the hairs are moving and telling you your body position. So we call them the semicircular canals. These get easily disturbed. When they get disturbed, we lose our balance. We become dizzy. If you've ever been on a ride like at Dollywood, that you get off the ride and you're dizzy, and you can't, your writing reflex is trying to find your equilibrium and the equilibrium and try to find your horizon, and it seems to keep moving, and it takes a while to you get balanced out again. And it's because these little hairs have been thrown around on the inside of the semicircular canals, and you are dizzy, off balance. It takes a while for that to calm down, and then you feel better afterwards, or you go throw up and you feel a lot better. So, okay. Now, if you notice again, this is all spongy bone right in here. So sound transmission works really well through that bone. Tympanic membrane, the ossicles, the auditory nerve. That is the cochlear branch of the auditory nerve. This is the vestibular branch over here. And those are the 3 semicircular canals. And that's the vestibule where the stoppase is going to go in through the, not go in, but send vibrations in through the oval window. And this gives you a pretty good idea of what this whole thing looks like. The semicircular canals are arrayed in 3 different directions. You have a vertical and 2 different types of horizontal directions, so that we can account for any kind of movement that we make. This is when we say, well, our inner ear got messed up. We got dizzy, because it happens. There are numerous types of disturbances that can affect the inner ear and affect our balance. So, you get an inner ear infection, and it can disturb the balance maintained by our equilibrium. Okay, and there's the cochlea right there. Looks like a snail. Stoppase, vestibule, And that was the semicircular canals there. And I just mentioned all that now. Semicircular canals, they depend on these hair cells, very similar to the hair cells inside the cochlea. They're in fluid and they vibrate. The vibrations, they function as mechanoreceptors, and their role is to vibrate and generate a graded potential that may reach threshold to tell you what's going on. So the cochlea works the same principle. It takes these hair cells that you see right there. They're the hair cells, very similar hair cells to what's in the semicircular canals. They vibrate. They vibrate their mechanoreceptors. They generate a graded potential, which if it hits threshold, we get an action potential. So now, okay, test for conduction deafness. Two types of deafness we encounter: conduction deafness and nerve deafness. Conduction deafness means we can't get the vibrations to the inner ear, to the cochlea. Okay, that's one type. Nerve deafness is the other type of deafness, where we can't get action potentials to move down the auditory nerve. And so one type can be helped with hearing aids. That's conduction deafness. We can do a workaround around the blockage. Perhaps the ossicles don't vibrate. Perhaps the tympanic membrane doesn't work. We can get around that conduction issue. If it's a nerve issue, there's nothing we can do, at least not right now. Now, we have a very simple test for this. We use a tuning fork. This is a tuning fork right here. You can see the tuning fork in the picture, but let me show you. This is a tuning fork. And if you know anything about tuning forks, they generate a musical note. In this case, it says that is a C. And if I strike this, you should be able to hear the sound on here from that. And all this is doing is vibrate. All the tuning fork is doing is vibrating. We probably can't see it vibrating, but it's vibrating and generating a note, a musical note, which is, a C. I don't know what a C sounds like, but it's a C. Okay. We can use this to test our hearing. If we take this tuning fork, and let me get my ear in view here. There's my ear. Somewhere. Okay, there's my ear. If I take the tuning fork and I hold it up next to my ear, I can hear the tone. You probably all hear it too. You all hear the tone, right? Okay, just say yes. So, thank you. Anyway, now the test for conduction deafness is to take the tuning fork and press it up against your mastoid process right here. As long as the vibrations are being transferred to the inner ear through the mastoid process, you can hear the sound. And it's gradually fading away. And it's still fading. But it's being passed through the spongy bone around the mastoid process to the inner ear. Now, when it fades out, and you can't hear it anymore this way, to determine if you can hear through the ear canal, take it and hold it up against your pinna. And if you can still hear it again, I don't know if you can or not, but still vibrating, I can hear it here, which tells me that I'm getting conduction through the auditory canal, through the ossicles, or through the mastoid bone into the inner ear. And so there is no issue with conduction. If there's a hearing problem, it has to do with the nerve. So, and it works. If anybody wants to play with this, I would encourage you to try it out. So, and it works. If you can hear the sound from both ways, pressing against your mastoid process till it goes away. So you hold it against your mastoid process until you no longer hear it, with the round end of it, and then like that, and then hold the two prongs up against your ear. If there's no conduction issues, you should be able to hear the note again here, because it's coming in now through the ear canal. It works both, that way it's showing that you have conduction going both ways. So, okay. So you can go ahead and try it. Yeah. Okay.
Sounds weird.
So we call this it's cleverly called the tuning fork test, so. Okay. Now, we are going to take a look at the sheep eyeball. And while the tuning fork is going around, I'm going to pass out trays.
I tried to catch it. Don't ignore. It's zero. Sugar. It's got artificial flavor. Right now, oh, when you went to try it.
Are you gonna cut? Okay.
Gonna do it.
You can stand. Yeah.
No.
Okay, now let me put my gloves on and get out the eyeballs. This, for those of you that are zooming, this is what our eyeballs look like. We have a package of eyeballs. They are CaroSafe Cal Eye plain bulk bag. Okay. So, get back to this. So now I know it says sheep eye on the slide. Sometimes we have sheep and sometimes we have cow, and they all look about the same.
Never think about that. Yeah, you want.
Now, you ever notice, those cookies you see at Christmas time, chestnuts?
No, don't ruin cookies.
Oh, come on.
Don't ruin the cookies, come on.
Okay, I won't say a word. I will not say a word. Yeah. Okay, now Looking at the eyeball, before we get into the cutting and slashing part, a couple things you should be able to see. First of all, you see a dark area on the front of the eyeball. And that dark area is actually the sclera. The sclera is the transparent portion of the eyeball. It doesn't look transparent, but that's what this area is. Let me point this out here on our model. This area right here, this is the cornea. This is the transparent portion in the middle of the sclera. The sclera is the white area, and the dark area that we see there is not the sclera. So what we're seeing on our slide, we see the cornea right there. The sclera is the white area around all this. Now it is transparent. It doesn't look like it's transparent, but you will see later on, either on the PowerPoints or on the eyeballs that are going to be cut up here, that it is a transparent structure. Should see a lot of fat around the edges of the eyeball, because our eyeballs are held in our socket, and our eyeballs have a lot of fat around them for energy and for padding. A person who has been ill for quite a long time will often appear to have their eyeballs sunken into their sockets, simply because a lot of the fat has been used up. You should also see a series of muscles around the eyeball. We have what are called intrinsic eye muscles. There should be 6 sets of these muscles all the way around to control the movement of the eyeball. Let me get an eyeball out here and I'll show you. Hey, you want to catch? Okay. Now let me clean my hand off before I put the camera on this. Let's bring this over here. Okay. So here's our eyeball right here. And this is the cornea right here in the eyeball. The fat layers are these yellow structures here, like right there is fat. These, this is a part of the muscle along the side here and along here. There's a nice set of muscles right there on the underside. We control the movement of our eyeballs in the socket by the movement of these muscles in here. And this structure, let me get some of the fat off here. You can see this better. Where do you get it open? The optic nerve, oh, here we go. This on the back side is the optic nerve. Now, if you look at it very carefully, I describe it as something about the size and shape of a mechanical pencil eraser. And the texture is about the same. If you find it and rub across it, you'll feel the same thing. And I expect you all to go ahead and look at that so you can know what I'm talking about. Okay, now. Okay, so there's our muscles and our fat and the cornea. We're not ready to cut yet, but I'll show you here in a second. The white matter is called the sclera. The sclera covers everything except the cornea. The conjunctiva is a layer of tissue which covers the entire sclera in the visible front. The conjunctiva protects the sclera, the white of the eye that we can see. So inflammation of the conjunctiva is called conjunctivitis or pink eye. So now, if you turn your eyeball around and look at the back side of it, you should see the structure there we call the optic nerve. This thing right here is the optic nerve. And if you push away the tissue, you should be able to find the optic nerve. It's okay, it's not going to bite you.
I know it's not.
It was looking at you.
I think it is looking at me.
Yeah, well, there, right there, is your optic nerve. See that?
Yeah.
And if you rub it across that, you'll feel that the nerve is much more rigid than any other portion of the eye.
Yeah, the lovely smell. It gets worse for references.
There's the nerve right there. Rub it across that. It won't hurt. Feel the difference? Feels like
Does
a
that
razor.
feel like a no, that doesn't feel
I was
like
gonna get through
razor. It feels like
scrambled eggs, I just didn't want to
a-
burn anything.
Of course, the thing is so wrong.
Would you stop burning food? Stop burning up
There's
food.
your optic nerve right there. Rub it
Really?
across
Oh, stop it.
Oh, God. It's never, dude. Real bad.
No.
It won't hurt. Yeah, right there. It's totally different. Moving around it. The best analogy is the pencil. Yeah. So, okay, so we have 4 pencil erasers and 1 mushroom. Now, the optic nerve, it consists of lots and lots of axons. You know, a nerve is made up of not just, you know, for a nerve, it's not just an axon. We have lots of axons in here, and this is a pretty large nerve going to the sheep brain or the cow brain. Now, the okay, a lot of fat around there, a lot of muscles, but that's the optic nerve. 2 other locations, we can see the optic nerve in both of these. This one on the right here, again, you can, when you push all the other tissue away, you can see the location of the optic nerve, and that penetrates into the back of the retina, to the middle of the retina. That's where the blind spot is, where we're going to be seeing the optic nerve leave the retina itself. Now, there's our optic nerve on both of these. So, and again, just in case you didn't notice it, that was the cornea. And the sclera. And the optic nerve on this side too. So now if you want to take your eyeball and cut it in half, and I want you to be very careful with your scalpel when you do this, but you're going to make a sagittal cut. Now, if you remember what a sagittal cut is, you will take the eyeball and you'll cut it in half down its midline. So like you would start it out like right here, like that. And then just like that. And then cut it in half all the way around. So now I will tell you the secret, the best way to do it is to stick your scalpel in and pop into the vitreous chamber and then take your scissors and cut. That's a whole lot easier than stabbing it all the way around with the scalpel. But you can do it either way, whatever works for you. So let me get this opened up for you.
Is it horizontal or wide cheeseburger? Okay, where do we cut this? That way or that way? If you know what you're doing either. What are we trying to be serious about this? We're gonna do this wrong.
There's no wrong when you do this, really.
So the fact that sagittal is kind of relative just to being in line with the eye. There's
Yeah.
no way you got to turn it around.
Yeah, no, just, you know, like lay it out flat like this and then cut it down the middle. You know, you're going to make a sagittal cut.
It looks like where you take a piece of a pineapple and bite into it.
Wait, so are we gonna head around there? I don't know.
Well, pineapple.
Oh,
Ew.
I don't like.
the
Oh, so we're supposed to be cutting this out.
so now what I've done,
Oh.
Oh,
if you take a look at the eyeball here,
Oh.
let's get the other camera, maybe that'll work better.
Don't slice it so it's like.
Okay,
my God.
There it ain't cutting. Don't keep slipping up the apron.
it ought to, if we're cutting open the eyeball, it should look something like this. What I've done here is I've made a cut right down the middle of the eyeball, a sagittal cut. I've cut through the cornea. I've cut through the front chamber and the back chamber. And if you look inside here, you can see the lens is about ready to drop out. There's the lens. I'm going to pop the lens out for you right there. Oh, that came out nicely. Here's our lens on the eyeball. And the lens is it made out of a crystalline, of a protein called crystalline. And it has it's not a perfect oval. It has sort of like a bowl-shaped structure in here. You can see it on here how it has sort of a flat surface and then a bowl shape, sort of like a Tupperware bowl, you know, a little really tiny Tupperware bowl. So that is the lens. And if we open this up now, we can see, let me get the rest of this cut through here. Now, this is the inside of our eyeball. Now okay. I'm gonna let everybody get caught up to that while I go ahead and talk a little more about what you should be seeing. Okay. Okay, so if you make a cut that looks like this, you should be able to see the lens, this large structure here in the middle is the lens. There's the cornea. You should have cut halfway through the cornea, and then you'll have the lens behind the cornea. There's the anterior chamber, the posterior chamber. There's the lens right there. Now, in front of the lens is the anterior chamber filled with fluid. The fluid is lacrimal fluid that in humans is released from the lacrimal gland to wash our eyeballs off, so that they can be clean of dust and dirt and debris and pathogens, because there's a certain antibacterial property of the lacrimal fluid. If that fluid accumulates in the anterior chamber and doesn't drain properly, it destroys the photoreceptors on the retina. This is the root cause of glaucoma, is increased pressure. This is the test that they do when they blow out a little puff of air at your eyeball when you're getting an eye exam. Now, you also notice that this structure right here is the iris, the pigmented portion of our eye, and that opening is the pupil. The pupil is simply the hole that you have to allow light to come in, to strike the lens, and then be focused on the retina. You've got the lens out, very good. So take a look at your lens and see if it doesn't have that sort of bowl-shaped structure I showed you on the one I have up here. Let's see, yeah, it's a very, very rigid structure now, but when while it is transparent in our eyes, it does have the ability to expand and contract as we accommodate for close and distance vision. So now, what else do we have on here? Okay. The vitreous chamber is the area behind the lens, and it is filled with a transparent jelly. The jelly gives the eyeball its shape. You know, so if you look inside your eyeball and see a bunch of clear jelly, that's what we call the vitreous humor. You got it open yet?
Yeah, got the lens.
Oh, you got the lens on, very good. Well, cut it into 2 pieces.
All the way from
All the way around, yeah, cut it into 2 pieces. Just cut it all in half, yeah.
Right here, this is.
That's the lens,
What you
yes.
pulled out is the
Right,
lens.
it looks just like this, the one I have up there. So
And then this is like, oh, okay, that's the
the
gem I knew, that's the lens.
Right.
That's the right way.
yeah, you did fine. It's not, you're cutting it all the way to you have 2 pieces. Oh, that's
Give me the scissors. I got
Okay. Oh, those are muscles. Muscles. On the belly there is called the Vitreous Humor. It's transparent and it sits between the lens and the retina. That thin brown layer right here. That's the retina.
You're not sick.
Oh, that's very good. Yeah. The one thing that we don't have, thousands of animals do, is this lower layer here. That's called Pituitary. And that's what makes our animals' eyes closed. It gives them a night vision that we don't have. It's so cool. We did. You know, it's incredibly pretty. So. Okay. So you've all seen the vitreous humor, that jelly in there. Okay, everybody's struggling to get it cut, but you know. Well, I know, but it's you just have to work at it. Okay. Okay. Well, while they're working, I'm going to finish cutting this, and then we'll take a look at it. Okay, well, this came out nice.
Like on the inside, this black piece under the
Mm-hmm.
membrane.
The black area is the what we call the choroid layer, and it's sort of like a light absorber for us. So. Now, to show you that the cornea really is transparent, I'm going to turn this around and let you take a look in here. And this should let you see it. Okay, now let's take a look here. If you look very carefully, you can see where the cornea is located. This is the cornea out here. Oops, there's the cornea right there. I've cut through half the cornea. Look at the inside of it. Come on, get in the picture. There we go. There, oh, there we go. That's that structure there. The opening is called the pupil. And we can see through the cornea is transparent. So we're actually seeing through that. Because look, you can see the probe. Let me get it lined up right there. You can see the probe through the cornea. So it's right there. So the cornea is letting light come through. And it goes, the pupil is this structure here, this opening. The pupil regulates light coming into the eye. Now, the pigmented portion of the eyeball is the iris, and the iris consists of smooth muscle that contracts in 2 different directions. The iris is what allows the pupil to open and shut, because the pupil is just a hole. But when we're looking at the iris here, we're looking at the colored portion of the eye. And let me take away some of the outer covering here, and I'll show you what the iris looks like. Well, here, I'll show you on the screen first, and then you can see what the iris looks like. There's our lens right in the middle. Now this dark band around the lens is called the ciliary body. And those are muscles. These are the muscles that allow for the lens to accommodate. So we can change the shape of the lens for close vision and for distance vision. And they are attached to the lens, and they'll pull it out or they'll compress it so we can look at things up close and look at things far away. Now, the other thing to see here is ciliary body. Now we're looking into this eyeball from the perspective of the lens, looking through the pupil, because this opening here is the pupil right there with the ciliary body around it. And now we're looking at it from the other side. We are in front. We are in the anterior chamber. We are looking at it from the perspective of the cornea looking in, and this brown structure there is the iris. And there's the pupil. Yeah. The thing that gives our eyes their color, the pigmented portion of our eyes, is the iris. You know, whether it's blue or green or brown or black or hazel. In the case of the sheep or the cow, they're probably brown eyes. So I guess they usually are brown eyes. Or maybe the preservative turns them into brown eyes. So now let's, I'll cut away some more of the front of the eyeball here. And so I can't expose the iris. Ooh, oh, this is nice. Now, the jelly, I pulled half the retina out with the jelly, and I've got to show you this on the camera because the retina will often stick to the vitreous humor. So this is what we're looking at right here. There's the jelly right there. There's the jelly. This is the vitreous humor, the jelly part of the eye. And on the back side of the jelly, this is the retina. And the retina, if I can slide it off, I will. But it's very, very thin, and it's prone to being damaged very easily. You know, you've ever heard of a person with a detached retina? The retina will let go from the choroid layer and have to be surgically put back. As you can see that the retina in this case came along with the jelly. You know, it's not supposed to do that. It's supposed to stay stuck to the wall back there. But this is the retina. And what you're seeing here in the retina, what these structures are probably axons leading to the optic nerve. So we see all along in here. Trying to get that there. There we go. Trying to get the retina to come off. Oh, there it comes. Yeah, now you can see how thin and transparent the retina is. This is the retina. This is part of the retina anyway that I pulled off of off the jelly. Now the other side is intact. And now notice here, let me get the jelly out of the other side. Well, no, I don't think I want. Okay. Now, the optic disc or the blind spot here in the retina is, if you look very carefully, let me see if I can get this visible, right there. So my thumb's in the way. Okay. That structure right there is the blind spot. Now, this isn't very as prominent as it usually is, but this is where all the axons in the optic, in the eye come together and then leave in the optic nerve, because here's the optic nerve right here. And so our blind spot is located right here where all the axons are feeding into that to go out to the optic nerve there. Now, let me show you on the PowerPoint what it looks like. Okay, pupil iris. Now, there's the vitreous humor. It's not unusual to get it to come out like that as a whole blob of jelly, you know. So, you know, like yeah, just like that. It's exactly what it comes out looking like. And you can see in the picture on the left that the eyeball, the lens, and the ciliary body are still attached to the vitreous humor. Even on the right-hand side, it's not as clear on the right-hand side. The ciliary body has been pretty much destroyed, but we do see some of the ligaments that are holding the lens in place. The lens is held right behind the anterior chamber by these ligaments. And then the ciliary body also holds the lens in place too. Most of the ciliary body has been destroyed, but you can still see where some of the ligaments are attached to it, to the lens. Now this next one, there we go. This covers everything. Here we see the lens sitting on top of the vitreous humor, the jelly. We can see the ciliary body wrapped around the lens. We see the retina, and there's this edge here, this raised edges here. Those are axons feeding into the optic disc or the blind spot, because the optic nerve is going to be just projecting through on the other side. So, and here we see a better picture of the blind spot. We see this up here is the blind spot. Now you've all probably noticed this blue pigmentation in your eye. Well, and it's very, very pretty. It's a very vivid iridescent blue. This is known as the tapetum lucidum. The tapetum lucidum is what allows animals to have incredible night vision. The tapetum lucidum amplifies the light rays that are striking on the inside, so that the eye, it's like a dog or a cat has like 10,000 times more ability to see at night than we can. That's why their eyes glow. You know, you see a rabbit with red eyes. It's not the rabbit from hell. It's just a rabbit with, you know, and you know, your dog or your cat or you see a cow across the street and the eyes glow. It's because light's bouncing off the tapetum lucidum in here. Different animals, the tapetum lucidums are different colors. Some are yellow, some are orange, some are red, some are gold. A lot depends. You know, red would be more of a highly vascularized retina, for example. But it all functions the same way. We don't have this. It would be very cool if we did, because then we could see great at night, but we can't. So, but that's that pigmentation portion you see there. That's the tapetum lucidum. The dark area is called the choroid coat. The choroid coat is the middle layer of the eyeball. The outer layer is the sclera. The choroid layer is in between, and then the retina is the inner layer. The choroid layer is a dark layer to absorb light rays. So you don't have extraneous light rays bouncing around the inside of the eyeball, bouncing off the retina. There's our retina. I think everybody got to see what their retina looked like. It's very, very thin. And it's very fragile. This is the first time I've ever had the retina really stick to the vitreous humor like it did, which is pretty cool the way it happened. But you can still see, you know, if you can pull your jelly out, you can see the jelly's inside the vitreous chamber. You can still see how it pulls the retina along with it. I've never seen it do that before, but that's pretty neat. Now, and there is the blind spot, the optic disc, where the optic nerve is leaving the eye. There are no photoreceptors located in that spot. So any light rays that strike the optic disc are not going to be detected. So that's why we have a blind spot there. And that is our eyeball. So now, if you want to keep playing with it for a few minutes, I'm going to clean up a little bit and then get ready to talk about the brain. So we don't have any brains to cut up. I'm sorry. So
I'm looking forward to it.
I know, so was I, but there's a shortage of brains on this campus. Said there's a shortage of brains. Or maybe there's a shortage of brains in this building. I don't know. But I have to know if you told Dr. Robinson what I said. Or were you afraid to?
I wasn't afraid to. I think we just got distracted. I could. I mean, I'm going to be in contact with him for a few days, so if you want, I can throw that in.
No, it's okay. He's such a scary person anyway, so, but he does get easily distracted, Oh, look, a cookie, So if you don't like what he's saying, just throw a cookie at him and he'll be okay. So. Now, I will say that this stuff is packed in like an alcohol-based solution. Yeah, because if they all come from Carolina, they're all packaged in alcohol, not formaldehyde. The other reason why I didn't get any brains out is the brains we have left are in a bucket of formaldehyde, and they stink, and formaldehyde's unpleasant. So, yeah. So, okay. Now let's take a look at the brain while we're here. The brain has the lobes of the brain are going to have the same name as the bones that are on top of them, and which of course gives name to the muscles which are on top of the bones. So we have a frontal lobe, a parietal lobe, and a occipital lobe, and a temporal lobe, which we won't see here for a second. The first thing you see with dealing with the brain is the cerebral hemispheres. These are the large structures on the brain. This is where where we are located. Most of what we are is in the cerebral hemisphere. And then you also see a large groove down the middle here. So the groove is called the longitudinal fissure. So longitudinal fissure divides the cerebral hemispheres into a right side and a left side. Our bodies are contralateral, meaning the right side of the brain controls the left side of our body, and the left side of the brain controls the right side of the body. There's very few places where we have a structure on one side and not on the other. I'll show you those in lecture on Thursday, where we have some speech centers which only show up on the left-hand side. There is the two cerebral hemispheres divided by the longitudinal fissure. There's the cerebellum, the second largest portion of the brain. Behind that is the spinal cord right there. And the spinal cord runs into the brainstem, and where the spinal cord begins and the medulla oblongata ends is very problematic. They just run together somewhere outside of the foramen magnum. The little grooves are called a sulcus. The sulcus become landmarks for us. We have a very large groove in the brain called the central sulcus, and that divides the frontal lobe from the parietal lobes. We have a series of ridges called gyruses. All these bumps are called a gyrus, and we use those as landmarks too. So for example, we have the central sulcus, and we can have the precentral gyrus, the ridge in front of the primary groove. Or we can have the post-central gyrus, the ridge behind the central groove. These ridges and grooves become landmarks for us. They're also there to increase the surface area of the brain. So it isn't just, well, we have ridges and grooves. Okay, fine. Well, why do we have it? Well, because we want to have a greater surface area on the brain. Now, frontal lobe, occipital lobe, and parietal lobe. There we go on the side. The only thing we can't see is the temporal lobe simply because it's a little further down over the side. So if we flip the brain over and look at the underside, we can see, of course, there's the spinal cord again. Now we can see the medulla oblongata. We can see the pons. The pons is a bridge. In fact, I think pons means bridge in Latin or something like that. It's a crossover. This is where spinal tracts coming up through the spinal cord are going to cross over, so that tracts from the left side of the spinal cord are going to cross over to the right side of the brain. And tracts on the right side are going to cross over to the left side because we have this contralateral control. So the pons is the structure above the medulla. The medulla has an awful lot of work to do for us with reflex centers and monitoring heart rate and blood pressure and breathing rate and stuff and swallowing. So the pons really doesn't get to do a whole lot except cross over the spinal nerves. In front of the pons is the pituitary gland. Now that's considered the master gland of the body. It is not the largest endocrine gland, but it releases 8 different hormones that act on our body. So when we get into AP2, we'll talk all about the role of the pituitary gland and the different hormones that come out of there and where they act. And if you remember from the Bones lab, the pituitary gland is found in the sella turcica, one of the most protected areas of the brain. Directly in front of this, well, actually, there's the temporal lobe off to the side. There is one of the optic nerves. We have an optic nerve coming from each eyeball. The optic nerve will split into 2 branches. And so each branch will come down. We see the left branch coming down there and the right branch on the other side. But at that point, they're going to split so that one branch, one split will go this way. And the one from this side it'll go straight back. It'll also cross over. This structure is known as the optic chiasma, where the optic nerves split and cross over. So each eyeball sends a portion of its optic nerve to both parts of the occipital lobe in the back. The left occipital lobe and the right occipital lobe each get information from the same eyeball. So you don't get left eyeball going to the left lobe and right eyeball going to the right lobe. You get the left eyeball going to the left and right occipital lobe, the right eyeball going to the left and right occipital lobe. That gives us binocular vision and allows us to see in 3 dimensions. So, because we have this crossing over here. Now, once we go into the optic chiasma, we cease being the optic nerve and we become the optic tract. So, but before we enter the central nervous system, we're still the nerve. So, now there is the olfactory bulb. The olfactory bulb in a sheep is really big. On a sheep brain. Now these sheep brains are about 6 inches long, and the olfactory bulb is over 1 inch long. Ours is not anywhere like that. Our olfactory bulbs are located directly above the cribriform plate up here at the ethmoid bone. The olfactory nerve branches come up from the nasal cavity. Our olfactory epithelial cells are located at the top of the nasal cavity on the inside. They pass up through the cribriform plate and they enter into the olfactory bulb. And then they go into the olfactory tract from the bulb as they go to the part of the brain where they get processed. So very large structure here for us. And so our olfactory nerves are not visible right now, but we definitely can see the olfactory tract here when we get some tissue removed in a second. Okay, and here we've removed some tissue. There's our spinal cord. This is the dura mater. I mentioned this in lecture today. This is connective tissue that surrounds the brain. It is the outer covering of the brain. It has the arachnoid layer and the pia mater or pia mater adhering to the surface of the brain. If you look carefully, you can just almost imagine that you see something here or some tissue like there and maybe there. That's the pia mater because it is stuck right to the surface of the brain itself. And you know, of course, they put the arrow where I see nothing, but you know, that's just me. So now, pituitary gland, again, temporal lobe. Now, this structure here is the optic chiasma. The optic nerves have been cut off. A lot of times when you get these specimens to look at, I've never seen one yet with the pituitary gland attached to it because it's always been, when they take it out of the sheep, you get what comes out with the brain. And so in 18 years, I've never seen a pituitary gland. And I've seen lots of optic chiasmas that have been just cut down to nothing here. So, so sometimes you get lucky and sometimes you don't. Now, but what you do see, there is the olfactory bulb on each side. And this large structure right up here is an olfactory tract. And if you look carefully, you can see another one right there. So these are very, very large structures that carry information back. And that makes sense because if you're dealing with a sheep or a cow, their sense of smell is incredible, much better than their sense, their vision. You know, sheep don't have great vision, but they sure do have a good sense of smell. They can smell water, they can smell predators, they can smell where the grasses are better to eat than other places. And so they can detect all that with their olfactory receptors. So they have a huge olfactory bulb. You know, we're lucky if we can, you know, if we can smell when supper's ready, you know, because we have a very tiny olfactory bulb. And yet we still can detect 10,000 different odors. And the sheep laugh at us instead of the cows, you know, because theirs are much better. That's what they're doing, you know, when you go by, they're laughing at you, you know, so just to let you know. So, okay. Mamillary body. This is the area that receives olfactory input. It's the only only sense that doesn't go through the thalamus. I mentioned in lecture today, the thalamus receives all sensory input. Everything except odors. Odors go through the hypothalamus and are passed through the mammillary body to get to the hypothalamus. So, yeah. And here's, we pushed the dura mater out of the way. There's our spinal cord, the medulla oblongata, the pons. You know, there's two large bumps on the brainstem. We're looking at the brainstem now. The bump furthest down towards the spinal cord is always the medulla. The one closer to the body of the brain is the pons. So first bump, second bump. Now this is the trigeminal nerve. We don't usually see that on these, but on humans, the trigeminal nerve is one of the 12 cranial nerves, and it has 3 branches. That's why we call it the trigeminal nerve. The 3 branches come across your face on each side. One innervates the area around the eyes, one innervates your upper jaw, the maxilla, and one innervates the lower jaw. So they're on, if I get this right, like this. When you have dental work done, you're usually going to be numbing or blocking the sodium channels, say it correctly, on either the maxilla or the mandible, depending on which part of your mouth you're having work done where you need anesthesia. The whole idea is block the sodium channels in the maxillary branch or the mandibular branch, and then the pain signals get to that point and stop. You don't stop generating pain signals, you just block them. Because your whole face goes numb. We've all been there. We've all had, I guess most of us have had dental work where we've been anesthetized, right?
Yeah, but why did you do it all the way back here then?
Well, I guess, you know.
I got shot all the way back there in that part.
Yeah, well, the shot always is worse than anything else. I hate the shot, you know. But if you've had dental work, you know that what they're trying to do is they're going to go back here, they're going to get the beginning of the branch. If it's in the lower jaw, they want to get where it splits, because it's going to have to split like this. So everything from here down on that side is completely numb.
This whole time my face was numb.
Yeah, that's exactly what it's supposed to do. Maybe he got a little further back than he wanted to, but or she wanted to. It doesn't matter. The whole idea is to me that the sticking the needle in to numb you up so you don't feel pain hurts more than anything else.
Yeah.
So and the what I always thought was so strange when I got at dental work was when they take that little ultraviolet device and they shine it in there. Like, what is that? You can't tell me that's going to do something, and it does. It cures the resin or whatever they're using in as the filling now. But the first time they ever did that, I thought, you got to be kidding me. What is this, like your kid's ray gun or something, from Star Wars or whatever, But it works, and it works very well. Anyway, trigeminal nerve, the mamillary body, the dura mater. That should be 1T, not 2. I didn't write this. So now, this is what I was talking about in lecture today about the corpora quadrigemina. The ability, well, you missed all that. So this is new stuff for you. So should I throw something at him to see if he can test it?
Yes.
Okay, good. So the corpora quadrigemina is a very important reflex center in the midbrain. It's on the dorsal surface of the midbrain. It's on the back side. The midbrain proper is on the other side. Now, the dorsal midbrain has 2 large lumps and 2 smaller lumps, or pedicles or projections, or whatever you want to call them. For reference, there's the occipital lobes. There is the pineal gland right there. There is the superior colliculus of the corpora quadrigemina. It's called superior because it's on top of the inferior colliculus. Now, this is a reflex center for vision. This is what allows you to track moving objects. It is also what is triggered by your peripheral vision. The rods that surround the retina in your eyeball are triggered by motion off to the side because they're very sensitive to that. They're on the edges of the retina. They don't get to do a whole lot during the day, but they're great at night. But they do pick up vision on the edge of your visual field. As they say, out of the corner of your eye, you detected somebody moving or something happening, and so you turn your head in the direction of the stimulus. It's a reflex. If there was a bright light suddenly appearing outside, we'd turn our head in that direction. If somebody walked by in the hallway, we'd probably turn our head in the direction if we noticed them. So it is a visual reflex center. We also use it when we're tracking things that are moving, like, the fact that you can catch that. Go ahead, yeah. The fact that you can catch that is because your corpora quadrigemina is tracking that. It's the same thing we use when we pull out in traffic. You come to an intersection and we all know what traffic's like around here at certain times of the day, and you're looking for a gap in traffic that is far enough apart so you're not going to get smacked, but not too close that you're going to get everybody excited when you pull out, So, or like my mother-in-law, nothing moving within a 5-mile radius before she pulls out. So, but that's okay. She's a safe driver other than that. So, but because we assess it, we're assessing that motion, Now the other area is the inferior colliculus of the corpora quadrigemina. This is where we turn our heads when we hear a sudden noise that we're not expecting. You don't have to sneak out. It's okay. So, well, that's okay. That's what we're talking about. So, the inferior colliculus, put the door stop in there so it won't lock. It's not outside. So, I used to have someone come by the classroom when we used to leave the doors open and take a big metal dissecting tray and slam it down out in the hall so everybody would turn their head like that and see what was going on. We can't do it anymore because we got to keep the doors locked, shut and locked. So, but it's a reflex center for sound. You turn your head in the direction of a sound to alert you as to what's going on. We call it the startle reflex. Someone calls your name, you turn your head in the direction of that. Maybe they're calling you, maybe they're not. You know, if you have a common name, then you know you run the risk of, hey, how are you doing? Like, oh, and they're talking to the person behind you. You know, you're doing the little head scratch thing. You know, it happens. But the sound thing is a reflex to protect us. You know, whether it's a police siren or an ambulance or a fire engine, or someone blows their horn at you, or you just hear something that doesn't sound right behind you or near you or something like that. You know, so these are two very important reflex centers. It's on the dorsal surface of the midbrain. So the midbrain is the top of the brainstem, and this is on the back side of the brainstem, on the top surface of the brainstem. So, and there, of course, there's the cerebellum. Now, later on, I'll show you in lecture that the pineal body releases a hormone called melatonin. Melatonin is what regulates our day-night cycle. It keeps us awake during the day and lets us sleep at night. It is based, the release of the hormone is based on visual cues we get from light. So right now, with being daylight, we're not releasing a lot of melatonin. As the day wears on and it gets darker, then once we start losing the visual cues for brightness, we'll start releasing more and we'll get tired. Think about in the winter when it's dark at 5 o'clock or 6 o'clock and you feel tired earlier than you do in the summertime. You know, summer it's like, hey, it's 9:30, it's still light, let's go do something. In winter, it's 9:30, it's dark, I want to go to bed. You know, think about it. It's the melatonin working for us so that we can keep our day-night cycle regulated, whether we want it or not. So, okay. Sagittal section of a brain. We see both gray matter and white matter. So we have myelinated axons in here too. We have unmyelinated axons. The most important areas of the human brain are unmyelinated, by the way. We'll see that in lecture 2. So a couple of things you see. First of all, right there is the optic chiasma. Now, this is a sagittal cut, so it's sort of hard to tell what you're looking at. But that's the optic chiasma. There's the pituitary gland. There's one of the cerebral hemispheres. You know, the cut was made, it looks like it was right down the middle of the longitudinal fissure here. And the dura mater. Now this structure here is called the corpus callosum. The corpus callosum is a set of fibers that run from one side of the brain to the other. What you're seeing is just a cross section of them, but they actually extend into both hemispheres. And that's how they're a set of fibers, communication fibers, that allow the left side of the brain to talk to the right side and the right side to talk to the left side, so that we stay in complete contact all the time. So the left hemisphere knows exactly what the right hemisphere is doing and vice versa. So, and in the middle of that corpus callosum is a ventricle, a chamber filled with cerebrospinal fluid. So corpus callosum right there. This is the cerebellum. The cerebellum contains both white matter and gray matter. The cerebellum, the organization of the white matter, they call it the arbor vitae, which means tree of life. When it was thought that maybe the center of life came from the cerebellum. What the cerebellum actually does is control our equilibrium and our balance. It's where we have our muscle memory stored. It's where we refine the actions of the cerebral cortex. This very thin layer right up here is called the cortex. And the term cortex is not unusual in a lot of organs. We'll talk about it a lot in in AP2. But the cortex, the cerebral cortex, is this outer 5 millimeters of brain is where we are located. Everything about us is in that outer 5 millimeters. It literally is like the icing on the cake. Everything underneath it is for association and processing. But everything about our personality, our intellect, our memories, our sense of right and wrong, all that are in the cortex. So, and it's all gray matter. Everything underneath it is usually white matter. So, okay, now, there's the thalamus. There are 2 thalami. They're paired. They're about the size, they're sort of egg-shaped. And that's where all sensory input from all the receptors all over our bodies, inside our bodies, come into the thalamus and get routed to the appropriate areas for integration and processing so that we can tell what's coming in. So we don't send an action potential from the eyeball to the center of processing for odor. So we know, so we can figure out what we're receiving. So, okay, same type of cut, but looking at it from the outside. There's the olfactory bulb, and you can even see the olfactory tract working its way back along here. There's the pons, there's the medulla, spinal cord, cerebellum, the cerebral hemisphere. Flip it over again. Spinal cord, pons, medulla, I always say that, medulla oblongata, and the pons. There's our thalamus, a better picture of the thalamus right there. Again, it's sort of like a little egg-shaped structure. They're paired. There's the corpus callosum, and now we can see the ventricle inside here, and that's filled with cerebrospinal fluid, and it's continuous with the ventricle surrounding the thalamus and continuous with the aqueduct that runs down here. And this is another ventricle here. The fornix is the junction of the both sides of the corpus callosum. Now, there's the pineal gland right there, and this is the corpora quadrigemina right behind it. The superior and inferior colliculus are right there. And let's see, okay. Now, so this opening in here is for the one of the ventricles. We'll see here, it's the lateral ventricle. There's the, right there in the corpus callosum, filled with spinal fluid, cerebrospinal fluid. It's continuous with the third ventricle, which surrounds the thalamus. That is continuous with the cerebral aqueduct, which runs down through here, all the way down to the fourth ventricle here. There's the cerebral aqueduct. And there's the fourth ventricle. And then there is a canal that runs down the spinal cord called the central canal. And the fourth ventricle leads into that. And that fluid then moves up and down from the spinal cord into this ventricle, into that canal or aqueduct, around the thalamus, up into the lateral ventricle, and then back again. About 5 or 6 ounces in the human brain. Not very much, just enough to support the brain and keep it supplied. Okay, and that was the central canal. And that's our brain. Okay. If you would like to keep your eyeballs, that's fine, but you don't have to. Just if you would dump them in the trash as you leave, that would be perfect. So, and I'll warn Herb that there's body parts in there. So, oh, just leave the tools in the tray and I'll take care of all that. I hope you enjoyed the eyeball. So I did. I don't, you know, like the rest of you, but I had a good time with it. Does anybody have any comments or questions on anything before I shut this down? Okay.